Hello everyone, and thank you for joining our today's VPHi webinar. I am Devan Sarwar, a PhD student in the Auckland Bioengineering Institute at the University of Auckland. The VPHi webinar series is a quarterly event organized by the VPHi Student Committee that provides a forum for access to senior community members and their expert competence for chiefly young scientists, but also to the VPH community as a whole. Today, we have the privilege to hear with us Professor Poul Nielsen and Dr. David Nickerson. Paul is affiliated with several groups within the University of Auckland. He is a principal investigator at the Auckland Bioengineering Institute, professor at the Department of Engineering Science, and honorary professor at the Liggins Institute. Paul's research focuses on using novel instrumentation, detailed computational models, and quantitative descriptions of physical processes to gain a better understanding of human physiology. Many of his projects couple mathematical modeling with innovative instrumentation to improve our ability to understand and interpret measurements of complex biological systems. Our another speaker, David, is an Aotearoa Foundation Fellow and Senior Research Fellow at the Auckland Bioengineering Institute, where he leads the Auckland Renal Physiome Project and helps develop tools and standards in support of reproducible computational physiology. David is an elected member of the CellML and ZML editorial boards, as well as being a combined coordinator. He also develops several software tools related to his work on model exchange, as well as being involved in several ABI infrastructural software projects. So in the first part of the webinar, Paul will discuss his experiences developing standards for encoding mathematical models to assist in their creation, reproducibility, and reuse. And in the second part of the webinar, David will provide an overview of the current state of specific standards, repositories, and software tools relevant to the VPH and the Physiome project. So welcome, Paul and David. Now I would like to invite Paul to start with his presentation.
Thank you, Dewan. As Dewan says, I'm Poul Nielsen, and both David and I will be talking about reproducibility and reuse in the virtual physiological human. But before we start, just to note that the three of us are actually in Auckland, New Zealand, and as you can see from the slide, this is, I guess, a depiction of what the globe looks like from 20,000 kilometers above Auckland, and you can see that we are a long way from anywhere apart from Australia and Antarctica, and the other view is from the other side of the world, where I guess where most of the participants are located. So physiology is the study of various organ systems at the higher level. In many cases in the ABI, we are interested in looking at these organ systems. In fact, we have various groups who are focusing on all 12 organ systems. But of course, that is just the beginning. The function of various organ systems rely on processes that are occurring on quite different spatial scales and temporal scales, ranging over a very, very wide range of scales, ranging from 10 to the minus 12 meters in the case of gene networks up to meters in the case of systems models of the entire human. And of course, the temporal scales are similarly wide-ranging, from 10 to the 9 seconds for the human lifetime down to at least 10 to the minus 6 seconds for molecular events. Not only that, but there is also a very large scale in the processes that occur over these various time and spatial scales. The information that we can derive at the organ system level is quite varied. as is the information that we can gather from organs and tissues down to the cellular and subcellular level. So the question is, how can we take advantage of all of these types of information, organize them in such a manner that we can get a better understanding of the processes that are occurring that makes up the human organism. Back in late 90s, that was the main question that was motivating the formation of the IUPS Physiome Project. It is fairly clear now, as was then, that the only way that we can really deal with this large quantity of information that we can obtain from various experimental measurements and imaging methodologies is to organize them in terms of mathematical models that are based upon physical principles. The human mind, of course, can deal with some of the integration, but there is no way that we can organize the information that we have, quantitative information that we have over these multiple levels, dealing with multiple physical processes without the help of mathematical models. That was really the main motivation for the formation of the IUPS Physiome Project, to take advantage of the mathematical modeling capabilities to put some order in the understanding that we have about the various organ systems at these multiple physical, temporal, and physical and temporal scales. So one of the problems that we have in proposing that models form the basis for our understanding of the human physiology is the inherent problems that we have in the publication process. In all cases of the publication process, the formulation of the model, the submission of the model to a journal, the refereeing of the models by experts, publication, typesetting, and the reading, the interpretation of these models, and then the subsequent translation into a program that is able to provide quantitative results to someone else, involves error-prone human intervention. This was the problem that we were facing in the late 90s when we were trying to exchange new models principally associated with cardiac physiology, but this is a problem in general, exchange models with our collaborators. All of these steps, because they involve human intervention, invariably lead to some degree of ambiguity of interpretation, and also errors creeping into the process. So that was part of the motivation we had then, to provide a mechanism that would allow us to share models of biological processes with our collaborators in a form that would bypass the majority of the human interventions, and hence the processes that can lead to errors creeping in. What we wanted was a way of describing models in terms of quantitative mathematical representations. We wanted a system that would allow the models to be reused, and more importantly, we wanted to have a way of describing models in terms of the mathematics that is in a form that is both human readable and also directly interpretable by computers. It just so happens around about that time when we were trying to tackle this problem, the systems biology community was also realizing that there were similar problems that they were facing. So there were two separate efforts that were built upon the Physiome community in the case of CellML and the systems biology community in the case of systems biology markup language, or SBML. Although the emphases were quite different, there was a significant overlap in the eventual solutions to this problem of being able to share models quantitative models in a way that provides a major reduction in the error-prone human intervention. So in the case of CellML, we wanted to include information about the model structure. That is, the topology of the models, how the various parts of models are related to each other. We wanted to have a mechanism that allowed us to describe the mathematics of the models in relatively high-level description of the processes. So, in other words, to be able to describe these models in terms of differential or integral or algebraic equations, not in terms of the algorithms that are used to solve such models. We also wanted a mechanism to allow us to include metadata, that is, additional information that is not strictly pertinent to the mathematical description, but can be very, very useful for describing information about the models that are useful to, for instance, discovering models about certain processes in certain species under certain conditions. So as a result of this, we devised this language, CellML, that includes the mathematics and metadata. All of this was based upon XML, which is a very general form of encoding information quantitatively about processes. We were keen to take advantage of existing standards. In particular, we were keen to use content MathML in order to describe the mathematics associated with the models, but also XML to provide the linking to, between various models, and RDF in order to provide some information about the metadata associated with models. So what I'll do here, these are fairly simple concepts. It seemed obvious to us in the late 90s that we would want to, leverage these existing technologies, relatively new technologies, and to build upon that a relatively simple set of constructs that would be able to provide us with the mechanisms to describe quantitative models, but to do so in a fairly simple way. So CellML is based upon fairly simple concepts. At the base of these concepts is something called components. It's really just a way of encapsulating some ideas, encapsulating some variables and the relationships between the variables, the units, and to do so in a way that provides a well-controlled way of providing an interface to other components. The other components can be connected together via connection facilities. Connections provide the mean for sharing information. They essentially take the interfaces from various components and link them together. An important aspect of the public interface and the units that are, the variables that are making up the components is the fact that we demand that all of these variables have an associated set of physical units. The idea behind this was to enable various groups to use quite different unit systems, but enforce unit consistency when components are shared between groups. There was also this idea of encapsulation. It was felt quite important that not only should we have the a flat component structure linked together by connections, but there should be some mechanisms for information hiding. And this was the main reason behind having both public interfaces and private interfaces. Private interfaces provide a connection or a linkage between components that provide some degree of hiding of structure beyond the public interface. Then at the top level, there is this concept of a model. It's really just a mechanism to provide a container for various components and their connections in a form that enables the models to be packaged together. Excuse me. And then beyond that, because we knew that we wanted to encourage the reuse of models, we provided this facility of being able to import models within other models. So the idea here was that groups would be publishing models that would form a useful basis for creating supermodels that are composed of several models from quite different sources. And then in order to provide a mechanism for encouraging this reuse of models, we set up a repository. At the time it was called the CellML model repository, but more recently, of course, it's been part of the Physiome efforts. So it is referred to as the Physiome Model Repository, or PMR. At the moment, we have quite a large number of well-curated models within the repository. Currently about 600 published models ranging over a wide extent of different physical processes, as you can see from the list here. And in fact, although we have a major emphasis on physiological models, we also include mechanical constitutive relationships, which is quite useful for being able to feed into distributed parameter models, as we'll get on to later. I should note that the SBML community have also got a similar repository, which is available from biomodels.net. The models that are available from the Physiome model repository are available at models.physiomeproject.org. Most of the models that we have included in the repository are reasonably well curated. We can give you an idea of the level of curation by having star tags associated with them. I will leave it to David later to explain why Miriam compliance is useful in this context. But suffice to say that we do have a very large number of models that are have associated publications that are freely available from the Physiome model repository, encoded in CellML and available for downloading and running and modifying and combining with other models. Okay, so most of the initial work that was done in the development of CellML was focused on cellular level models. In particular, if we are looking at cellular and subcellular processes, a good example of this is excitation contraction coupling at the subcellular level, how, for instance, the various ions, concentrations and the gate states allow calcium to flood into the actin-myosin region and enable contraction process occurring within, say, skeletal and cardiac muscle. Of course, these processes are well studied, and there have been many models of the aspects of the processes that occur at this level proposed over the years. A good example of this is the Lura-Rudy-2 model, published in 1994, which consists of about 20 coupled nonlinear ODEs. It is this type of model that we were initially targeting CellML to be able to describe, partly because the only mechanism that we had for exchanging such models was in the form of MATLAB or Fortran or some other encoding of the models that assumed a particular algorithm for integrating, for solving such models. But those processes that occur at the cellular level, although they are able to be well described using lumped parameter models, that is, we don't have any concept of spatial variations in any of the variables, for instance, the concentration of ions, are inherently useful for distributed parameter models. And it is at this low level where the lumped parameter descriptions are able to be described using CellML that can feed into the higher level descriptions where spatial variations become quite important. Although CellML is focused mainly on the lumped parameter descriptions, that is, systems of ordinary differential equations, these are nevertheless quite useful to be feeding into distributed parameter models, that is, partial differential equations, as you would often find, for instance, in models of large deformation mechanics of the heart. So we devised a process to take these CellML models to be able to be plugged into the distributed parameter models through a compilation process that provided a sort of a plug and play mechanism for using the low-level process descriptions into high-level models. What I'm showing here is this process of coupling the lumped parameter descriptions at the cellular level into a distributed parameter mechanistic description of cardiac mechanics. In fact, this is a model that David Nickerson, the second speaker, devised maybe a decade ago at least. But it is still a useful description of how the CellML processes can be conveniently and efficiently used in these higher level descriptions of distributed parameter processes. So the other sort of main aim that we had with CellML was to provide a mechanism for enabling researchers to take models of physical processes and be able to plug and play those. A good example of this is this form of encapsulation is the various levels of complexity associated with the calcium current associated with calcium gates at the cellular level. So, for instance, Bieler-Reuter 1977 model has a relatively simple calcium ion description, gating description, which was improved by Luo-Rudy in 1994, and then further improved by Jaffrey Rice Winslow in 1998. Each of these models can represent to certain degrees the processes that give rise to the calcium current. The nice thing about the CellML descriptions is that essentially you can choose whichever one of these models you want to use in order to best describe the process, the overall process that you are trying to represent, depending on the level of fidelity. So it's this plug and play idea that was central to the development of CellML. CellML 1.1 has this idea of importing models to create supermodels. This was a minor tweak to the CellML, the initial CellML 1.0, which essentially required you to cut and paste models in order to create supermodels. But the import element provides a mechanism that allows you to do this automatically, essentially providing the route to creating more complicated models from much simpler ones. An example of how this is achieved is given in the following set of slides here, where 3 models of different aspects of cardiac contraction. So, for instance, Pandit et al. Model of cardiac action potential, which is a medium complexity model consisting of, I think, 30 or so coupled ODEs, which has a representation in the Physiome model repository and can be downloaded to be solved by various applications, for instance, OpenCore. My apologies to Alan Garney here. I have a very, very old screenshot of Core, which has got much better facilities nowadays and much nicer graphics. But the idea here was to combine that Pandit et al. Model with the Hinch et al. Model of calcium-induced calcium release, which is similarly described in the Physiome model repository and can be downloaded to run under OpenCore, with a Niederer et al. Model of myofilament mechanics. Here again, the model is downloadable and runnable via OpenCore through the Physiome model repository. So, Terkildsen about 10 years ago spent quite a bit of time combining these 3 models, taking advantage of the CellML import facility to create a supermodel which integrated these 3 physiological processes into a single model published in 2008. And you can see here from the screen, this is really quite a complicated model that is integrating three quite separate processes associated with different physical phenomena. And once again, via the import element, the import facilities, this model is downloadable from the Physiome model repository in a form that is essentially a linking together of the three constituent models. So that really begs the question then, how should we use CellML effectively? Well, perhaps the best way to approach CellML is to, rather than use CellML to describe already, functional models that integrate various processes, perhaps a better way to think of CellML is to actually decompose those models into reusable functional units. Every reasonable model that has been published describing relatively complicated physical phenomena is made up of functional units that can be reused. So, for instance, the Pandit model contains many reusable units. The concept here that we are trying to pursue is to look at the published models that we have, to find what are the reusable components, what are the constituents, to decompose those down into reusable components, and then to build the published models up from those reusable components, allowing researchers who are interested in looking at variations, different combinations of models, to be able to do so relatively easily based upon the published models in the basic models published in the Physiome model repository. So, as I mentioned earlier, CellML 1.1 allows model to be decomposed into many smaller files by aggregating submodels. Now that begs a question: how do we manage this level of complexity? Because any model is going to be made up of many different component files. And that was one of the main motivations that we had for developing the Physiome repository. This was initially based upon a distributed version control system, allowing many different developers to work on a model or various aspects of a model independently, interacting only insofar as they needed to in order to create a supermodel or a larger model that is based upon those components that they were developing. So the distributed version control system that forms the basis of the Physiome model repository is I think a very nice way for a group of modelers to be able to work together to construct models, to create models, allowing multiple versions of shared components without ever having to rely on a centralized server. We are still, feeling our way through what is required for solving such large-scale problems, and it's fairly clear that we can't rely simply on the mathematical and topological description of the mathematical, representation of the models. We need to be able to provide additional information about the species, for instance, about the conditions under which the processes are occurring, about the physical principles that underlie the processes that we're trying to describe. And it is for that reason that we introduced a linking mechanism that associates CellML entities to ontologies. the ontologies provide us with a mechanism for including such information in a well-codified form that provides the ability to not only search for particular characteristics, but to perform logical operations on such characteristics. And essentially to provide a codified way of representing our knowledge about systems beyond the mathematical descriptions. I will jump over that. So in summary, before I hand over to David, we developed CellML to be a versatile format to define high-level representations of mathematical models. We have also created a model repository that provides a mechanism for public access to a wide range of curated biological models that describe many different types of processes. And we have provided a mechanism that allows us to link to ontologies to provide additional information about the models that are go beyond the mathematical descriptions. A lot of this information and explanations can be found on the CellML project website indicated below here. And just to note that we have an annual get-together, a workshop that is conducted every year, typically during from March to May, on a lovely island just a short distance from downtown Auckland called Waiheke. If you're interested in participating or learning more about this, you can check the CellML website under the Community Events Workshop page. So what I'll do now is hand over to David.
There we go. So thanks, Paul, and thanks to the VPH Institute Student Committee for asking us to talk. So just in the last few minutes, I was just going to quickly run through a few updates, I guess, to various things that are going on in this sort of community that you may or may not have seen before. So the first one really is just to mention that there's this combined network, the Computational Modeling and Biology Network, where a bunch of us working on standards like CellML and SPML sort of get together a few times a year just to make sure we're not overlapping too much, and try and developing these standards in a way that they're all orthogonal. We can use common technologies where possible, and things like that. So in Combine, there's these core standards that sort of form the basis of the network. And so they span roughly 3 different types of representation. So there's the idea of just representing knowledge directly, where we have BioPax, which is a biological pathway exchange sort of format, where you can encode sort of network topologies and connectivity, but not necessarily quantitative models. And there's also the synthetic biology open language, which is for representing in synthetic biology things like DNA sequences and that sort of thing, where you might be developing a sequence that you then send off to be made and assembled in various sort of expression systems. And then there's standards for visual representation. So here we have the systems biology graphical notation, which provides sort of standard graphical representation for how you can draw various types of network topologies and sort of define function, where they're trying to provide the equivalent of a sort of electrical circuit diagram for systems biology, and gradually expanding in scope to try and cover some of the more sort of physiological models that we might deal with CellML. And then SBOL have their own visual language for defining their sort of DNA assemblies. And within Combine, we're trying to work together to try and develop a common way to describe these things. And then there's the ones we're probably more familiar with in terms of models and their analysis. So here you see systems biology markup language, CellML. The neurophysiology community have their standard NeuroML, and then also the simulation experiment description markup language, which is a way, is a language that describes how you can take mathematical models in whatever format and apply some sort of algorithm to them and produce some outputs and any post-processing you might want to do to essentially generate those final outputs that you might put in a paper or see in a paper. And then alongside these core standards, you know, as a community, we can develop these common sort of infrastructure and other things that are useful to all of us, regardless of which particular standard or community you might be coming from. So one of the more recent ones is this idea of a project, what we call a combined archive, which is a way to package up everything you need into a sort of reproducible unit that you can easily hand around and exchange between software tools. For those that have heard of research objects, this is a very similar idea, and in fact, working with the research objects people, you can sort of translate combined archives and research objects fairly easily nowadays. And then various sort of infrastructure things that we use for the annotations. There's this identifiers.org effort from the EBI in Cambridge to try and provide a common framework for providing persistent identifiers, making sure they're always resolvable. And then various controlled vocabularies and ontologies where we need to define new vocabularies or ontologies that maybe don't already exist, or just making sure people know which ontologies they should use. So those resources are all on that website listed there. And another one nowadays that's becoming quite popular is this idea of the FAIR guiding principles for scientific data, but that obviously also applies to models as data as well. And this is the idea that everything should be findable, accessible, interoperable, and reusable. And for FAIR, it's particularly that this should be true for both people and machines. So not only should people, you know, using your web browser or Google be able to find and access things, but you want software tools and agents to be able to do the same sort of discovery and reuse. And this is where it starts to become important that we can describe our models and data using these sort of common vocabularies and ontologies and the metadata that Paul was mentioned earlier. And in terms of FAIR, there's this great website, fairsharing.org, or I guess it's a catalog sort of thing where you can go and you can find all sorts of information about all sorts of standards and databases and you know, operating procedures and things like that. So this is a good place to go and look for particular standards and see what's out there and what people recommend, and see if you can get any help. It sort of points you to the right people to ask questions or mailing lists to go to and things like that. It's quite useful. So now, Paul's already mentioned PMR, the model repository that we have that started with CellML. So I just wanted to quickly give you a brief overview. As Paul's already mentioned, you know, the core concept in PMR is that everything's in a workspace. Each workspace is a version-controlled repository, and you can, you know, as an owner of a workspace, you can control who has access to it. You can make them public once we just check that it's safe to do so and things like that. But that revision of the history of the workspace is there. Every version is persistently identifiable, so you can always go back to every version. And then in PMR, you can sort of take a particular revision of a workspace and do some processing of it to produce a pretty sort of web presentation. And we call those exposures. And then also more and more, what you need to do is take the annotations from those workspaces and put them into standard RDF or semantic web sort of knowledge bases so that you can find them and use them to do cool things. So some of the recent additions in PMR, so we are now starting to collect these sort of libraries of reusable modules that Paul was talking about. And here you can see we've got this currently small but ever-growing collection of protein modules where we take, provide models of specific transport proteins for different membrane types of things. And so they're available. And for those, you can see, hopefully, that this particular one of sodium hydrogen exchanger. And you can see not only this sort of base model, but a bunch of sort of supplementary models and simulation experiments that show the range of behavior of this particular model. You can see at the top, there's these sort of vocabulary terms that define what this model is, where it's located, links to various databases and things like that, to find out more information about it. And then what we're doing more and more now is, for these models in PMR, you can now directly launch the model into OpenCore, the desktop application. So as long as you have that installed, clicking that launch button will fire up OpenCore. And depending on what exactly it is, you may see either the model or a simulation experiment. So here it's providing the model, so you can now jump in and see the model, directly browse the equations and the math, parameter values, annotations, things like that, and then go on to perform simulations and explore the behavior of the model. And another thing you can do is you can then, when it's available, if you put the little bit of extra information in your workspace, then PMR is able to generate a combined archive for your workspace. And so that then gives you this nice little archive file. It's essentially just a zip file, which you can then pass to different tools and get the same, reproduce the same simulations. So here you can download that archive, and here we're showing the CellML web tools and OpenCore, and if you put that archive into either of those tools, you can hopefully see that you get the same results in the simulations. And then just to show with OpenCore now, it's working directly with the repository. So you no longer have to learn how to use Git in order to put content into the repository. You can do it all within OpenCore. So here on the left-hand side, you can see there's, if you have a reasonably new version of OpenCore, you can see there's a PMR workspaces window, and that's showing you essentially all the workspaces that you own in the repository, and you can get little, icons that tell you when things have changed or not changed, and then it's really simple to just click a button. It prompts you for a log message, and then it will synchronize directly into PMR for you with no sign of a Git client whatsoever. And then part of what it does is it will interact with any instance of PMR. So we currently have 3 different instances of PMR running that you can use. There's the main one at models.physiomeproject.org, but then we also have a sort of testing one at staging, and then a teaching one that we use for tutorials and things. So in OpenCore, you can easily just choose the instance that you want to point at, and then log into that particular instance of the repository, depending on if you just want to test things out or if you're doing real serious work and you want to make sure it stays there forever. Now, finally, we also have color. So in OpenCore, if you've used it before, you might have seen that you always had graphs that were the same color for everything, which made it a bit confusing. And now in probably one of the most recent snapshots of OpenCore, you can start to configure, which plots are which color and things like that, which has been a huge improvement. So if you want to have a play, it's definitely worth checking out. And then finally for OpenCore, this idea that you can now, if you load your model, you set up your simulations and your graphs and things like that. But if you want to share them with someone in a way that they can reproduce them, you want to generate either a CellML file, which just as that sort of recipe, telling people what they need to do with the model to produce those outputs, or a combined archive, which is that zip file that you can then share with people to give them what they want to be able to reproduce a particular simulation experiment or collection of experiments. And very briefly, just following on from what Paul talked about in terms of annotation, this picture is just trying to give you that idea where, in the top part, you can, give your variables in your model or your components, names that you may understand, but they may not make sense to a lot of people. And so then it's hard to know if you have 2 or 3 different models. If you use the same names or similar names, you might be able to connect things, but then if you have a different name, you don't always know how to connect it when you're trying to reuse a model. So this idea, if you link your variable names or variables to these vocabularies, which unambiguously define the biology that underlies what you're trying to model, then it makes it easy for software tools to now automatically connect these things together. So for example, the model that Paul showed before of the sort of electrophysiology, calcium and mechanics, there's a tool SemGen with the link there, and that using annotated versions of those models, we can now sort of do that model merging and integration pretty much automatically, which works really well. But of course, once you have models with these annotations in them, you can start to do more. And so here, just to show briefly work that Dewan's actually doing, you can have tools that will then do sort of smarter querying and discovery of what's in the repository. So you can have queries like, show me all the models that have something to do with the flux of sodium and things like that, and it'll go away and find, not just particular variable names, but now looking at the biology and the definition. And then once you can do that, you can find multiple things that maybe have something to do with the flux of sodium and see how they are semantically related. And this helps in terms of whether or not you might want to, reuse a particular model. If there's no overlap, or if there's some sort of point of overlap between 2 different models, then it helps you as the user decide whether or not to reuse a particular model. And then you can take this to the extreme where you start to now build up, essentially here epithelial transport models based on pulling in these little individual transporter models. And just quickly go through. So now trying to encourage people to put in a little bit of extra effort to make their models available in these annotated standard formats. We've recently launched this Physiome journal to provide a mechanism to essentially publish these curated and annotated models to hopefully encourage people to do it. We're partnering with Figshare to publish this, so we use the Figshare platform to make these available. And the idea is that these Physiome papers are always associated with a primary article, which is your traditional article where you're publishing a new model or experimental paper, and that goes through the normal peer review process that would then check the model and the science is okay. But then associated with that, you could then publish the model, encoded in CellML or SPML, or perhaps even MATLAB if you really wanted to, along with the annotations and curation information that will let people be able to discover it and reuse it. And then this is all done open access and with a low additional cost on top of your traditional sort of publication. And then finally, just as you start to build up collections of these models, not just in CellML, but in sort of larger scale models and these distributed sort of field type models, you can then build these web sort of base portals for exploring the information that these repositories contain and use that sort of annotated semantic information to be able to link all these things together to make it possible to build, rather simply these sort of web interfaces that connect things and let users browse and explore and discover the knowledge that's contained in one or potentially many repositories that are available. So I think that's it, what I was going to talk about.
Thank you very much, Paul and David, for your presentation. Now I would like to move on to question and answer session. Dear audience, please feel free to ask questions if you have microphone, or you could type your questions. So in the meantime, I would like to address first question to David Nickerson. So David, how to know which is the appropriate standard to use for your model?
So it's not always easy. So if it's like Combiner trying to help provide guidance as to which standards to use, there's the biosharing.org sort of catalog that I showed briefly, which will list them all, but doesn't really provide any guidance into which ones to use. For the sorts of modeling that we do, there's another catalog. I don't know how to show it, but if you Google for NormSys, it's from an institute in Heidelberg. There you find a nice little portal that says, where you can query what standard to use based on what you're trying to do, at least for the modeling standards. So things like the combined core standards and various other related standards that works well. And I think gradually we'll have to start providing similar resources for things like experimental data and things like that.
Thank you. I have another questions to Poul Nielsen. So why both mathematics and physics are important to build the virtual physiological human?
Well, the way that processes work within the human body is based upon the physics. We have, in many cases, a good idea of the physics associated with, for instance, diffusion or chemical interactions or electrophysical interactions, and that knowledge is usually able to be well described using mathematical relationships. That is why we have based our sort of quantitative descriptions of physical processes within the CellML language on mathematics, because it is the best way in which we can provide a quantitative relationship between the various variables of interest.
Yeah, thank you. I have one more questions to David Nickerson. So how do you link different organ models, for example, linking with heart and lungs in cellular level with CellML?
So it really depends what you're trying to do with your model. Certainly within CellML itself, you can easily connect various sort of lump parameter type models together. We have models in the repository that do that. Probably the more interesting thing is when you start to have something like a circulatory model, where you might have a spatial model of the circulation tree, and then you can look at how the different organs at the sort of terminal ends of the various branches of that arterial tree interact. And so depending on your software, you can sort of link those things together. Certainly we can do that in CMIS, and other tools I know of will let you do that. We define the sort of spatial models in some software and then link in, I think as Paul was describing, the CellML models in a sort of plug and play fashion.
Yeah, thank you. So Cora is asking that I lead the biomedical informatics group at ABI, and we are using the same semantic annotation framework to link computational models to real-world healthcare data using latest health informatics standards.
Okay, not really a question, but just a comment.
No, it's not a question. I can see that there are two raised hands. Two people raised their hands. Okay, so the first one, I guess, from Nima. Hi all, that's a nice talk. I was wondering, how can we connect enterocyte with other compartment like blood in order to make a composite model? And if we do that, how can we validate those models?
So the way that models are linked is through common variables. I'm sorry, I didn't pick up what the first process was, but
Enterosite.
right. You know, for instance, if they are interacting via, you know, shear forces in the blood, then that would be the mechanism by which the two models would be interacting. That would be essentially the way in which you would, in the lumped parameter case, provide the mechanism for linking those two models of different processes together. But first of all, of course, you need to identify through which variables the processes are going to interact.
Nima said thank you. Thanks.
Thank you.
Any other questions?
So maybe just to follow up on Corey's comment, just to mention that, as I think you might have put in the chat box, that, you know, once you have these models that are semantically annotated, so you're linking the mathematical model to the biology, it makes it possible now to start doing these types of things where you can link between the sort of computational models that we have and the clinical sort of health records and clinical informatics world. And there, the question is that we use very different terminologies in our typical modeling realm to what they do in the clinic. And how we connect those things together is sort of the question that we're trying to work on, or one of the questions we're trying to address in current work.
Yeah, Corey replies with thanks.
Yeah, I think we can close the session.
Thank you, Paul, and thank you, David, and thank you, everyone.
Great. Thank you again. Bye-bye.
Bye.
Bye-bye.